Welcome to Chapter 2 of our series on uveitis and ocular inflammation. In the first chapter, we grounded ourselves in innate immunity, the fast, hardwired defenses that recognize conserved molecular patterns. Today, we shift to the other half of the immune system, adaptive immunity, and specifically immunization and the immune effectors that drive intraocular inflammation. This is where the story becomes personal to the pathogen, because adaptive immunity learns, it remembers. And importantly for us as clinicians, it can turn against the eye's own tissues. Over the next hour, we'll walk the immune response arc from antigen capture through lymphocyte processing to the effector mechanisms that actually cause tissue damage. And we'll anchor each concept in real ocular disease, from sympathetic ophthalmia to Fuchs uveitis syndrome. Keep the eye in mind throughout. Every pathway we discuss eventually shows up at the slit lamp.
Let's start with the big picture, the themes that will recur on every slide. The defining trait of adaptive immunity is antigen specificity. Rather than reacting to broad molecular patterns the way innate immunity does, it produces a learned response tailored to individual antigens. The system is constantly sampling antigenic epitopes and performing one critical task, distinguishing self from non-self. When it recognizes an antigen, it activates lymphocytes, chiefly T and B cells, and crucially, it lays down immunologic memory. Adaptive responses come in three flavors: antibody-mediated, cell-mediated, or coordinated by T lymphocytes, or a combination of both. And here's the clinical hook that ties this whole chapter to your practice. T helper responses, particularly the Th1, Th2, and Th17 pathways, drive the immunopathology of many intraocular inflammatory diseases. Hold on to those three subsets. We'll return to them repeatedly.
Before we can talk about recognition, we need precise vocabulary. An antigen is any substance the immune system recognizes. But the immune system doesn't see the whole molecule. It sees an epitope, the specific portion of that antigen to which an antibody or receptor binds. This distinction matters because a single complex three-dimensional protein may display multiple recognizable epitopes, while other regions stay completely invisible to the immune system. And epitopes exist on both self and foreign tissues, which is exactly why autoimmunity is possible. Unlike innate immunity's focus on conserved patterns, adaptive immunity targets highly variable, specific epitopes. Its job is to sample those epitopes continuously, sort self from non-self, and eliminate what's foreign. To organize the rest of this lecture, we'll use a framework called the immune response arc, divided into three phases: afferent, processing, and effector. Everything that follows hangs on that scaffold.
This diagram summarizes the immune response arc from antigen exposure to the generation of effector and memory cells. The process begins when a stimulus, or antigen, is encountered in peripheral tissue. Macrophages and other antigen-presenting cells recognize and phagocytose the antigen, then process it for presentation. These antigen-presenting cells migrate through afferent lymphatic pathways to a regional lymph node. Within the lymph node, the processed antigen is presented to T and B lymphocytes, initiating their activation, proliferation, and differentiation. The resulting immune response generates effector T and B lymphocytes that can act against the offending stimulus, as well as memory lymphocytes that provide a faster response with future exposure. The key concept is that antigen recognition begins in the peripheral tissue, but organization of the adaptive response occurs primarily within lymphoid tissue before immune effector cells are deployed.
Let's enter the arc at the beginning, the afferent phase. This is the initial recognition, transport, processing, and presentation of antigen. It starts when antigen-presenting cells, or APCs, bind antigen at peripheral sites. Once they receive stimulatory signals, complement is a classic example, they phagocytose that antigen and then migrate through afferent lymphatics to the regional lymph nodes. Inside the APC, within endocytic vesicles, enzymes chop the ingested proteins into small peptide fragments, roughly 7 to 11 amino acids long. Those peptides then load into grooves on HLA molecules displayed on the APC surface. The resulting peptide HLA complex is what gets presented to T lymphocytes, which read it through their T cell receptor, assisted by either the CD4 or CD8 co-receptor. That handshake, peptide, HLA, and T cell receptor, is the fundamental unit of adaptive recognition, and we'll unpack each part next.
Now, why does it matter which HLA molecules you happen to carry? Because HLA molecules differ in which peptide fragments they can physically bind and present. That means your individual HLA type sets the menu of antigens that can ever be shown to your T cells. This isn't abstract genetics. Specific HLA alleles are among the most important susceptibility factors for several forms of uveitis. Think of HLA-B27 and acute anterior uveitis, or HLA-A29 and birdshot retinochoroiditis. Those associations trace directly back to this presentation biology. One quick terminology note that trips people up. HLA molecules belong to the major histocompatibility complex, or MHC, family. MHC is the general term used across species. HLA, human leukocyte antigen, is simply the human-specific terminology. When you read the literature, treat them as the same system. Next, we'll separate the two functional classes of HLA because they present very different antigens to very different T cells.
Let's take HLA class 1 first. These are the HLA-A, HLA-B, and HLA-C molecules, and they present antigen to CD8 positive cytotoxic T cells. The key feature of class 1 is where it's expressed, on nearly all nucleated cells in the body. And it samples the internal world of the cell. It primarily presents peptides synthesized inside that host cell. Under normal conditions, this is silent because presentation of ordinary self-proteins produces no immune response. That's self-tolerance working as intended. The system springs to life when it presents altered self-antigens. Viral peptides, tumor antigens, or mutated proteins can all activate cytotoxic immunity. But notice the double edge here. The same events, viral infection, neoplasia, genetic mutation, can also break tolerance and drive autoimmunity by triggering inappropriate responses against host proteins. That tension between defending the cell and attacking self runs through much of ocular inflammatory disease. This figure illustrates the class 1 pathway in action. Picture a nucleated cell. Remember, that's almost any cell in the body, synthesizing protein internally. Some of that protein is degraded, loaded onto HLA class 1 molecules, and displayed on the surface, where a CD8 positive cytotoxic T cell inspects it through its T cell receptor. When the peptide displayed is normal cell, the CD8 cell moves on. When the peptide is viral, tumor-derived, or mutated, that recognition licenses the cytotoxic cell to kill the presenting cell. This is why class 1 is often described as a window into the cell's interior. It lets the immune system audit what a cell is manufacturing, without the cell having to be a professional immune cell at all. Contrast this in your mind with the class 2 pathway we're about to see, which handles antigens taken up from outside the cell. The distinction of inside versus outside is the single most useful way to keep the two classes straight.
Now, the counterpart, HLA class 2. These are the HLA-DR, DP, and DQ molecules, and they present antigen to CD4 positive helper T lymphocytes. A defining rule: CD4 T cells only recognize peptide antigen when it's presented on a class 2 molecule. No class 2, no CD4 recognition. Unlike class 1's near universal expression, class 2 is restricted to specialized professional antigen-presenting cells, dendritic cells, macrophages, and B-lymphocytes, especially within lymph nodes. And these class 2 dependent APCs are specialists at a particular job, processing extracellular antigens they've acquired by phagocytosis. So the typical targets here are things from outside the cell, bacterial and fungal proteins, for instance. That's the clean division to remember. Class 1 surveys peptides made inside the cell for CD8 cells, while class 2 displays peptides scavenged from outside the cell for CD4 cells. Those CD4 helpers, as we'll see, are the master coordinators of the entire adaptive response. Here we see the class 2 pathway diagrammed. A professional antigen-presenting cell, a dendritic cell, macrophage, or B-lymphocyte, engulfs extracellular material, say a bacterial protein, into an endocytic vesicle. Enzymes fragment it. The peptides load onto HLA class II molecules, and the complex is displayed on the surface for a CD4 positive helper T cell to inspect. Notice how different this is from the previous figure. There, the antigen came from inside the cell, and the audience was a killer CD8 cell. Here, the antigen came from outside, and the audience is a helper CD4 cell that will orchestrate a response rather than directly kill. Keep in mind that only these specialized cells express class II constitutively. Most body cells cannot present to CD4 helpers. This restriction is precisely what concentrates helper cell activation in the lymph nodes, where APCs and naive lymphocytes converge. That convergence is where the ARC's next phase, processing, takes place.
We've captured and presented the antigen. Now we enter the processing phase, where an antigenic stimulus is converted into an organized immune response. Location matters here. Processing happens primarily within the lymph nodes and the spleen, the secondary lymphoid organs built for exactly this purpose. The central characters are naive B and T lymphocytes, cells that have never yet encountered their specific antigen. When antigen meets the one naive lymphocyte whose receptor fits it, that cell undergoes priming. Priming then progresses to full activation, which has two components you should commit to memory. Clonal proliferation, the cell divides to build an army of identical specificity, and differentiation into functional immune cells. That pairing of proliferation plus differentiation is the engine of adaptive immunity. It's how a system that starts with a handful of specific cells generates a response large and specialized enough to clear a pathogen. Over the next slides, we'll dissect exactly how helper T cells get primed.
Let's zoom in on the requirements for T cell processing, because the details here explain a lot of downstream disease. The principal cell in immune processing is the CD4 positive helper T lymphocyte. For it to recognize antigen, three components must come together into what's called the trimolecular complex: an HLA class II molecule, the processed antigenic peptide sitting in it, and the T cell receptor reading them. The CD4 co-receptor stabilizes that interaction between the APC's HLA peptide complex and the T cell receptor, and it amplifies signaling. Successful recognition produces priming, a partial activation. Primed T cells then acquire new functional capacities. They begin to divide, they synthesize cytokines, and they express adhesion and co-stimulatory molecules. One cytokine deserves special mention: interleukin-2. IL-2 is especially important for sustaining T cell activation and differentiation through autocrine stimulation, meaning the cell secretes IL-2 and responds to its own signal. That self-reinforcing loop keeps the response going once it's started.
This figure returns us to the class II APC, now in the context of processing. Picture the trimolecular complex we just described, rendered visually. The HLA class II molecule presenting its peptide, the T cell receptor engaging from the CD4 cell, and the CD4 co-receptor clasping the complex to stabilize and strengthen the signal. What this image emphasizes is that recognition alone isn't the whole story. The primed T cell that emerges from this interaction is being licensed to divide, to secrete cytokines, and to express new surface molecules. So think of this synapse between APC and helper T cell as the decision point of the entire adaptive response. Everything downstream, which cytokines dominate, which antibody classes get made, whether the response protects the eye or attacks it, is shaped at this junction. On the next slide, we'll see how the local cytokine environment at this very moment steers the naive helper cell toward one of several distinct fates. So, a helper T cell has been primed. What does it become? Before priming, CD4 T cells are uncommitted. We call them TH0 cells, blank slates in a sense. The cytokines present in the local environment at the moment of activation then direct differentiation down one of several paths: TH1, TH2, TH17, or regulatory T cells, TREGs. Each of these subtypes is defined by two things: a specific set of transcription factors that lock in its identity, and a characteristic cytokine profile that it will secrete. And those cytokine patterns are not academic. They determine everything about the subsequent response: how antigen is processed further, what class of antibody the B cells produce, and which cell-mediated effector mechanisms come into play. This is arguably the most important branch point in the whole lecture. Over the next 4 slides, we'll take each subset in turn, Th1, Th2, Th17, and TREG, and connect each to the ocular diseases it drives.
This diagram gives us the family portrait of helper T-cell differentiation, and it's worth pausing. At the top sits the uncommitted Th0 cell. Radiating out from it are the polarizing pathways, each labeled by the cytokines that drive it and the signature cytokines it will produce. Trace the branches. One arm leads to Th1 under the influence of IL-12, another to Th2, a third to Th17, driven by IL-6, IL-23, and their partners, and a fourth to regulatory T cells. Notice that these fates are cross-regulatory. Pushing toward one subset often actively suppresses another, which we'll see explicitly with Th1 and Th2. As I walk through the individual subsets on the coming slides, keep returning to this figure in your mind's eye. Uveitis and scleritis, sympathetic ophthalmia, ocular toxicariosis, Behçet's disease, each maps onto a particular branch of this tree. Understanding the branch point is understanding the disease mechanism.
Let's take the 2 classic subsets together because they define each other by opposition. Th1 cells are driven by IL-12, and their mission is eliminating intracellular pathogens. They mediate cell-mediated immunity and delayed hypersensitivity, and they secrete interferon gamma, IL-2, TNF-alpha, and TNF-beta. Th2 cells, by contrast, are stimulated by IL-2 and IL-4, and they specialize in clearing extracellular pathogens, but they also drive allergic disease, including asthma. Their signature cytokines are IL-4, IL-5, and IL-13. Now here's the elegant part. Th1 and Th2 cross-regulate each other. Interferon gamma, the Th1 cytokine, inhibits Th2 responses. IL-4, the Th2 cytokine, inhibits Th1. So the 2 arms act like a seesaw, each suppressing the other, which is why immune responses often commit strongly to one polarity. Keep this Th1 versus Th2 balance in mind. When we reach sympathetic ophthalmia and ocular toxicariosis, you'll see these 2 arms driving opposite patterns of eye disease.
Th17 cells are the newer member of the family, and for uveitis specialists, they may be the most important. They defend against extracellular bacteria and fungi, particularly at mucosal surfaces, and they're driven by a distinctive cocktail: IL-6, IL-21, IL-23, and TGF-beta. They produce IL-17, IL-21, IL-22, and TNF-alpha, and their differentiation is locked in by the transcription factors ROR gamma T and ROR alpha. Now look at the clinical list because it's striking. Dysregulated IL-17 and IL-22 signaling is implicated in psoriasis, rheumatoid arthritis, multiple sclerosis, and inflammatory bowel disease, in Sjogren's syndrome, Behçet disease, and systemic lupus, and, right in our wheelhouse, in uveitis and scleritis. This is why the Th17 axis has become such a prominent therapeutic target. When you hear about IL-17 or IL-23 pathway inhibitors in inflammatory disease, this is the biology they're aimed at. Th17 is where a great deal of modern immunomodulatory therapy is now focused.
The 4th fate is the peacekeeper, the regulatory T cell, or Treg. Where the other subsets drive inflammation, Tregs restrain it. They're stimulated by IL-2 and TGF-beta, and their job is maintaining peripheral tolerance to self-antigens. In practice, that means preventing autoimmune disease, limiting chronic inflammation, and actively suppressing autoreactive T cells that escaped earlier checkpoints. You can identify them by their marker profile, CD4, CD25, and the master transcription factor, FOXP3. Their suppressive muscle comes partly from the cytokines TGF-beta and IL-10. Here's the concept to carry forward. Immune health is a balance. When Treg-mediated suppression holds the upper hand, self-tolerance is maintained. But when the balance tips toward the pro-inflammatory pathways, too little suppression, too much Th1 or Th17 drive. That imbalance may underlie uveitis and other immune-mediated diseases. Much of the eye's remarkable immune privilege, which we'll return to, depends on keeping this regulatory arm dominant.
We've met all four subsets, so what actually decides which one a naive helper cell becomes? Three factors converge at the moment of activation. First, the pre-existing cytokines in the local microenvironment, the chemical context the cell is bathed in. Second, the nature and quantity of the antigen itself. And third, the type of antigen-presenting cell involved. A concrete example ties it together. Macrophage-derived IL-12 preferentially promotes a Th1 response, which is exactly why intracellular infections, handled by macrophages, tend to polarize toward Th1, and the consequences ripple outward. The resulting T-cell subtype shapes downstream B-cell antibody synthesis and the cellular effector responses that follow. So this isn't a random choice. It's a context-dependent decision that the immune system tunes to the threat. That principle, that the local environment programs the response, is also why the eye's specialized microenvironment can shape and sometimes misdirect intraocular immunity. Now let's follow the helper cell's influence over to the B-lymphocyte.
One of the most important jobs a helper T-cell has is activating B-lymphocytes. So let's turn to the B-cell side of processing. B-cells produce antibodies, or immunoglobulins, and each antibody binds the very antigenic epitope that stimulated its production. The specific antigen binding site is called the paratope, and it sits on the antibody's Fab region. Remember that term, because it'll recur when we dissect antibody structure. Naive B-cells display surface IgM and IgD, which serve as their B-cell antigen receptors. Here's a key contrast with T-cells. B-cells can recognize epitopes on intact, unprocessed antigen. They don't need an APC to chew it up first. That's a fundamental difference in how the two lymphocyte lineages see the world. But recognition alone isn't enough for a full response. Antigen receptor stimulation, followed by helper T-cell interaction, is what drives B-cell proliferation and differentiation. This is the T-B collaboration at the heart of antibody responses.
Once a B-cell is activated, it transforms. It divides, expresses accessory molecules, ramps up to produce large quantities of antibody, and ultimately differentiates into antibody-secreting plasma cells, the dedicated antibody factories. But the most clinically consequential trick is class switching. Activated B-cells can swap their initial IgM for another immunoglobulin class, IgG, IgA, or IgE. Crucially, class switching changes the immunoglobulin heavy chain while preserving the antigen specificity. The paratope stays the same, only the constant region changes. That means the antibody keeps recognizing the same target, but gains entirely new effector functions. And helper T-cell cytokines control which class gets made. Interferon gamma promotes switching from IgM to IgG1, while IL-4 promotes switching from IgM to IgE. Notice how that maps on to the Th subsets. Th1's interferon gamma pushing toward IgG, Th2's IL-4 pushing toward IgE. This is the molecular link between helper polarization and antibody function, and it explains why different diseases feature different antibody classes. We've captured antigen and processed it into an army of specific cells and antibodies. Now the payoff, the effector phase, whose job is to eliminate or neutralize the foreign antigen. There are two major antigen-specific effector pathways, T lymphocyte-mediated responses and B lymphocyte antibody-mediated responses. Alongside those, a set of non-T, non-B effector cells contributes, natural killer cells, lymphokine-activated killer cells, and other killer cells, cells that do damage without antigen-specific receptors of their own. Now, recall the round trip I flagged back at the ARC overview. Maximum effector activity generally requires two antigen exposures. The first is priming and activation in the lymph node. The second is re-stimulation out in the peripheral tissue, where the antigen originated. That two-hit requirement isn't a technicality. It's the mechanistic basis of the difference between primary and secondary immune responses, and it's exactly what a delayed hypersensitivity skin test exploits. Let's now look at each effector arm in turn.
Let's detail the T-cell effector arm, which splits into two functional types. First, delayed hypersensitivity T-cells. These usually express CD4, and they work by releasing cytokines, interferon gamma and TNF-beta, that recruit and activate other cells, especially macrophages. This is the engine behind granuloma formation and the tuberculin skin reaction, and we'll see it repeatedly in ocular disease. Second, cytotoxic T lymphocytes. These express CD8, and they're direct killers. They destroy virus-infected host cells and tumor cells on contact. Together, these two responses provide antigen-specific cell-mediated defense. One arm marshals the broader inflammatory response through cytokines. The other performs targeted assassination of compromised cells. Notice how cleanly this maps onto the HLA classes we covered earlier. CD4 delayed hypersensitivity cells reading class 2, CD8 cytotoxic cells reading class 1. Keep both types in view, because when we reach sympathetic ophthalmia, cytotoxic lymphocytes, and NK cells later in the lecture, we'll be expanding on exactly these mechanisms. Now, the antibody arm of the effector phase. B-cell effector function is carried out not by the B cells directly, but by the antibodies their plasma cell descendants secrete. Those antibodies drain into the efferent lymph and then enter the venous circulation, spreading throughout the body to find their target. Once an antibody binds its specific epitope, it can do two main things. First, it can promote opsonization and phagocytosis, essentially coding the target so that phagocytes recognize and engulf it. Second, it can activate complement through the classical pathway, unleashing that cascade's lytic and inflammatory power onto the target. Together, these mechanisms contribute to neutralizing and eliminating the foreign antigen. Notice the theme. The antibody itself is a targeting device, but much of its destructive effect is delegated to phagocytes, to complement, and, as we'll see later, to effector cells bearing Fc receptors. That principle of antibody as targeting tag will return when we discuss ADCC and eosinophil-mediated injury in the eye. This figure summarizes the immune response arc by showing how the adaptive response travels between peripheral tissue and the lymph node. At the tissue side on the left, antigen-presenting cells, particularly dendritic cells, capture antigen and migrate through the afferent lymphatic system to the draining lymph node. Within the lymph node, antigen presentation activates and expands antigen-specific lymphocytes. These differentiate into the three major adaptive effector populations shown here: cytotoxic T lymphocytes, delayed hypersensitivity CD4 T lymphocytes, and B lymphocytes that become antibody-producing plasma cells. The activated cells then leave the lymph node through the efferent pathway, enter the circulation, and return to the peripheral tissue. There, cytotoxic T cells directly kill antigen-bearing cells. CD4 T cells recruit and activate other inflammatory cells, and plasma cells produce antibodies that bind the target. The key point is that the tissue is both where the response begins and where it is ultimately completed. Here is the feature that truly sets adaptive immunity apart: immunological memory. Everything we've discussed so far, the innate system does in some form, but memory is uniquely adaptive. After an initial antigen exposure, a subset of antigen-specific lymphocytes doesn't die off. It persists as memory cells. The consequence is profound. On re-exposure to the same antigen, the system mounts a far more rapid and effective secondary response than it managed the first time. And this isn't just theoretical elegance. It's the basis of the single most successful intervention in the history of medicine. Protective immunization, vaccination, is the classic clinical application of immunological memory. We deliberately install memory against a pathogen so the body is primed before it ever meets the real thing. Over the next several slides, we'll dissect exactly how this works: the sluggish primary response, the formation of memory cells, and the swift, robust secondary response, and then ground it all in a tuberculosis example that illuminates ocular disease.
Let's examine why the first encounter with an antigen is so slow. The primary response occurs during that initial exposure, and it faces a numbers problem. Antigen-specific lymphocytes are initially rare, roughly 1 in 100,000 B cells, and about 1 in 10,000 T cells carry the receptor for any given antigen. So the system must first find those rare cells. The naive lymphocyte has to encounter appropriately presented antigen and become activated. And because activation begins from a resting state, building the machinery from scratch, the primary response typically takes several days to ramp up. That lag is why a novel infection can make you sick before immunity kicks in. But once activation occurs, the payoff begins. Activated lymphocytes undergo clonal expansion, dramatically multiplying the number of antigen-responsive T and B cells. That expansion converts a rare specificity into a substantial population, and as we'll see next, some of those expanded cells are set aside as memory.
So what happens to all those expanded lymphocytes once the threat is cleared? After antigen is removed, most activated cells return to a resting state, but they are fundamentally changed. They're no longer naive, they are memory cells, and that distinction is everything. The speed difference is dramatic. Whereas naive cells needed days, these memory cells can be reactivated within roughly 12 to 24 hours of re-exposure. They're also better positioned. Memory lymphocytes express increased adhesion molecules, including integrins, and that heightened adhesion promotes homing and migration into target tissues. The memory cells physically station themselves where they'll be needed. The result is an expanded antigen-specific population that persists at the very sites where future antigen exposure is likely. Put those pieces together, more cells, faster activation, better tissue positioning, and you have the cellular basis for the rapid, powerful secondary response we'll describe next. This is also, incidentally, why chronic ocular infections can sustain a persistent local pool of primed lymphocytes. Now we reach the payoff of memory, the secondary or anamnestic response, the reaction to a second or subsequent exposure to the same antigen. It's dramatically faster and more robust than the primary response, and for 3 concrete reasons. First, far more antigen-specific lymphocytes are already present, thanks to the clonal expansion we discussed. Second, memory cells activate rapidly, hours rather than days. And third, antibody class switching has already occurred, so the system starts with better tools. That third point has real clinical texture. Primary responses tend to produce IgM, a large pentameric molecule that penetrates peripheral tissues poorly, precisely because of its size. Class-switched antibodies, especially IgG, enter tissues far more readily, or can be produced locally, and immediately bind antigen. One more clinically important note. Persistent low-level antigen, as in mycobacterial infection, can maintain chronic stimulation of T and B lymphocytes, effectively keeping the secondary response smoldering. That sets up our tuberculosis case perfectly.
Let's make all of this concrete with tuberculosis, tracing the primary response right through our 3-phase arc. In the afferent phase, alveolar macrophages in the lung ingest Mycobacterium tuberculosis and transport the organisms to the hilar lymph nodes, antigen moving inward, exactly as the arc predicts. In the processing phase, T and B lymphocytes are primed in those nodes, and the resulting proliferation is what produces the hilar lymph node enlargement you can see on imaging. Then the effector phase. Primed T cells recirculate back to the infected lung and recognize macrophage-associated bacterial antigens. Their cytokines activate nearby macrophages, which differentiate into epithelioid cells and fuse into multinucleated giant cells. This is what builds the caseating granuloma, the histological hallmark of TB. And critically, some of those activated T cells persist as memory T lymphocytes. Hold on to that last point, because those very memory cells are what the tuberculin skin test reactivates, which is exactly where we're headed next.
Now watch those memory cells in action, because the tuberculin skin test is a beautiful demonstration of a secondary T-cell mediated response. When purified protein derivative, PPD, is injected into the skin, dermal macrophages take it up and migrate to the draining lymph nodes, where they reactivate the memory T-cells left over from the primary response. Those reactivated T-cells return to the dermis and encounter antigen again at the injection site. There's the two-hit, priming then re-stimulation pattern we keep emphasizing. Their cytokine release recruits additional lymphocytes and monocytes and promotes fibrin deposition, and within 24 to 72 hours, this produces the characteristic indurated skin lesion you palpate and measure. This reaction is the tuberculin form of delayed type hypersensitivity, a pure, visible readout of cell-mediated memory. Keep this delayed hypersensitivity mechanism firmly in mind, because it's the same fundamental process that drives granulomatous uveitis and sympathetic ophthalmia later in this lecture.
Having covered the arc in memory, we now turn to classifying how adaptive immunity actually causes inflammation and injury. The historical framework is the Gell and Coombs classification, which describes 4 immune-mediated inflammatory mechanisms. Type 1 is anaphylactic, or immediate, hypersensitivity. Type 2 is antibody-dependent cellular cytotoxicity. Type 3 is immune complex-mediated reactions, and Type 4 is cell-mediated reactions. This classification is still worth knowing because it remains useful for interpreting older immunology literature. You'll encounter these Roman numerals constantly. That said, for practical purposes, adaptive effector responses are more cleanly grouped into 3 categories, and this is the scheme the rest of the lecture will follow. First, antibody-mediated responses. Second, lymphocyte-mediated responses, including delayed hypersensitivity and cytotoxic T-cell activity. And third, combined antibody and cellular mechanisms. Keep this 3-part map handy, because it's the table of contents for everything that comes next. We'll start with the antibody-mediated arm and its structural foundation, the immunoglobulin molecule itself. To understand antibody function, we have to start with antibody architecture. There are 5 major immunoglobulin isotypes: IgM, IgG, IgA, IgE, and IgD. 2 of these are further subdivided, IgG into IgG1 through IgG4, and IgA into IgA1 and IgA2. Now, the basic building block. A single antibody monomer is built from 4 covalently linked glycoprotein chains, 2 identical light chains, either kappa or lambda, and 2 identical heavy chains. Here's the key organizing principle. It's the heavy chain type that determines the antibody's isotype and subclass. A monomer weighs in around 150 to 180,000 Daltons. But not all isotypes stay as monomers. IgM assembles into pentamers or even hexamers, which explains its large size and poor tissue penetration, while secretory IgA commonly forms dimers, suiting it for mucosal defense. Keep this scaffold in mind, because in the next slides we'll divide the antibody into its functional regions, Fa and Fc, and see how structure dictates function.
Let's divide the antibody into its 2 functional territories. Each antibody has 2 Fab regions, and these contain the variable antigen binding domains, the business end for recognition. The specific antigen binding site within the Fab region is the paratope, the same term we introduced with B cells. So Fab equals recognition. The other territory is the Fc region, formed by the heavy chain constant domains. In this is where effector function lives. Fc-mediated functions include binding to receptors on effector cells, fixing complement, and interacting with secretory transport components. 2 concrete examples make this vivid. IgG3 strongly activates complement. In IgA, binds a secretory component that lets it be transported across epithelium into secretions, such as tears, directly relevant to ocular surface immunity. So the mental model is a 2-part molecule. Fab decides what the antibody grabs, Fc decides what happens next. That division of labor, targeting versus action, is fundamental, and it's why the same antigen can trigger very different outcomes depending on isotype.
Let's turn that structural insight into a functional principle.
Immunoglobulin isotypes differ in the effector mechanisms they activate. The Fc region, remember, does the work, and each isotype's Fc behaves differently. Consider complement. IgM and IgG3 are effective activators of complement, whereas IgG4 does not effectively activate it at all, so switching class can switch inflammation on or off. Consider transport. IgA is uniquely carried into mucosal secretions through its binding to the secretory component. That's what puts protective antibody into tears and other surface fluids. Now, the punchline, and it's the crux of this whole section. Two antibodies may recognize exactly the same antigen, yet produce entirely different inflammatory effects, simply because they belong to different isotypes. Same paratope, same target, different Fc, different consequence. This is why, clinically, it isn't enough to know that an antibody response exists. The class of that response shapes whether you see complement-driven damage, mucosal protection, or allergic reactivity. Next, we'll add some essential terminology around antibody clonality. This table compares the 5 major immunoglobulin isotypes by abundance, location, and function. IgG is the predominant serum antibody, accounting for roughly 3/4 of circulating immunoglobulin. It functions in neutralization, complement activation, agglutination, and antibody-dependent cellular cytotoxicity, and it is the only major isotype that crosses the placenta. IgA is the main antibody of mucosal secretions and contributes to protection at epithelial surfaces through neutralization and immune exclusion. IgM is primarily intravascular and is especially important early in the immune response, where it efficiently agglutinates antigen and activates the classical complement pathway. IgE is present at very low serum levels, but is bound to mast cells and drives mast cell degranulation in allergic responses. IgD is also present at low levels and functions mainly as an antigen receptor on B lymphocytes. The key point is that antibody class determines where the immunoglobulin is found and which effector mechanisms it can activate. Now, some terminology that shows up constantly in the biologics era. Each individual B lymphocyte produces a unique Fab region, recognizing one specific antigenic configuration, one cell, one specificity. When that cell undergoes clonal expansion, it yields a population of identical B cells, all recognizing the same epitope. A population derived from a single B cell clone is, by definition, monoclonal. This is exactly the principle behind therapeutic monoclonal antibodies, engineered uniform antibodies against a single target. And you'll recognize the examples: infliximab, adalimumab, and rituximab, all workhorses in ocular inflammatory disease. Now contrast that with what the body does naturally. Most natural immune responses are polyclonal because multiple B-cell clones each recognize different epitopes on the same antigen, producing a diverse antibody mixture. So the distinction is, monoclonal means one clone, one epitope, one uniform antibody, the pharmaceutical product. Polyclonal means many clones, many epitopes, the physiologic response. That contrast sets up the next slide, where antibodies themselves become antigens.
Here's a concept with real consequences for how our biologic drugs perform. Because antibodies are themselves proteins, they can function as antigens. The immune system can raise antibodies against antibodies. The antigenic determinants located on an antibody are called epitopes. Keep the pairing straight. Epitopes are antigenic sites on foreign molecules, whereas epitopes are antigenic sites on antibodies. Antibodies raised against these are anti-idiotypic antibodies, and they do two things. Physiologically, they can regulate immune responses through feedback, but clinically, they can neutralize therapeutic biologic antibodies, and that's the problem of anti-drug antibodies. Take infliximab, a chimeric monoclonal antibody against TNF-alpha used in some forms of uveitis. Patients can develop anti-idiotypic antibodies against it, and these may reduce infliximab's efficacy by blocking its very TNF-alpha binding site. This is the molecular reason a biologic can lose effectiveness over time, and why we monitor for immunogenicity. It's a direct bridge from basic immunology to a decision you make at the bedside.
Up to now, we've largely discussed antibodies produced systemically, but the eye teaches us to think locally. Persistent antigenic stimulation can recruit B lymphocytes directly into the affected tissue. With chronic stimulation, those infiltrating B cells may differentiate right there in the tissue into plasma cells, and those plasma cells then produce antibodies locally aimed at the inciting antigen, antibody manufactured on-site rather than delivered from the bloodstream. This has a powerful diagnostic implication. Demonstrating local antibody production can support a diagnosis when a specific infectious antigen is suspected. If you can show that antibody against a particular pathogen is being made inside the eye at levels that can't be explained by leakage from serum, you have compelling evidence that the pathogen is driving the intraocular disease. This principle of local intraocular antibody synthesis is the foundation of a specific diagnostic tool we'll examine next, the Goldman-Whitmer coefficient, and its elegant application in Fuchs' uveitis syndrome.
Let's put local antibody production to work diagnostically. The Goldman-Whitmer coefficient is a tool for demonstrating intraocular production of pathogen-specific antibody. Conceptually, it compares the proportion of pathogen-specific antibody in the aqueous humor against that in serum. A high ratio means the antibody is being made inside the eye rather than merely leaking in. The classic application is Fuchs' uveitis syndrome. In affected eyes, the aqueous humor can contain markedly elevated rubella-specific IgG relative to serum. The numbers from Quinton and Reiber are striking. A mean Goldman-Whitmer coefficient of 20.6 in eyes with Fuchs' uveitis syndrome versus less than 1.5 in controls. That enormous difference supports a strong association between rubella virus and Fuchs' uveitis syndrome, a genuinely important etiologic insight, and the test earns its keep clinically precisely when intraocular antibody production is present, despite limited systemic evidence of active infection. When the eye knows something, the blood doesn't. It's local immunology turned into a diagnostic answer.
Local B-cell activity has a darker side too. When persistent tissue antigen combines with local B-cell infiltration and ongoing antibody production, the result can be a chronic Arthus reaction. The classic Arthus reaction is an immune complex phenomenon, antigen and antibody meeting in tissue and depositing as complexes that incite inflammation. And when it becomes chronic, it leaves a characteristic histologic signature. You may see lymphocytic infiltration, plasma cell infiltration, and even granulomatous inflammation in the affected tissue. This matters beyond the eye. This same mechanism may contribute to chronic autoimmune diseases characterized by pathogenic antibody formation, with rheumatoid arthritis as the prime example. So notice how the threads connect. The local plasma cells we discussed for diagnosis in Fuchs' uveitis can, in another context, become drivers of chronic antibody-mediated tissue damage. That completes our tour of the antibody-mediated arm. Now we pivot to the second major category from our three-part scheme, the lymphocyte-mediated effector responses, beginning with delayed hypersensitivity.
We now enter the lymphocyte-mediated arm, and its prototype is delayed hypersensitivity, DH, a quintessential T-cell mediated inflammatory response that's especially prominent during secondary responses. Let's trace its choreography. Previously primed CD4 T cells leave the lymph nodes and home to tissues where antigen persists. There, restimulation requires the specific antigen presented on HLA class II expressing APCs, the trimolecular complex again, now out in the tissue. Once restimulated, the activated T cells release cytokines that recruit and activate macrophages and other nonspecific leukocytes, amplifying a small, specific trigger into a substantial local inflammatory reaction. And here's the origin of the name. The reaction is called delayed because it typically reaches maximal intensity 12 to 48 hours after antigen exposure, the time needed for cells to traffic in and activate. You've already seen this exact mechanism in the tuberculin skin test. Now we'll see how the Th1 and Th2 flavors of delayed hypersensitivity produce distinct patterns of ocular disease. Delayed hypersensitivity comes in different flavors depending on the helper subset driving it, and the Th1 version is the classic one.
Th1 cells produce interferon gamma and TNF-alpha, which activate macrophages and amplify inflammation. Interferon gamma is so central to this that it's also known as macrophage activating factor, a name worth remembering because macrophage activation is the heart of Th1-mediated DH. Look at what this pathway is responsible for. The classic PPD skin test reaction we already dissected, immunity against intracellular pathogens, including mycobacteria and pneumocystis, responses to fungal infection, severe T-cell-mediated autoimmune disease, and chronic transplant rejection. That's a sweeping portfolio, and it's unified by macrophage activation and granulomatous inflammation. For our purposes, keep this Th1-DH pattern front of mind, because it's precisely the mechanism we'll invoke for sympathetic ophthalmia, a Th1-driven granulomatous panuveitis. But first, its mirror image, the Th2 version of delayed hypersensitivity, which produces a very different kind of tissue reaction.
Now, the mirror image, Th2-mediated delayed hypersensitivity, which looks quite different from its Th1 counterpart. Th2 cells produce IL-4, IL-5, and related cytokines, and these steer the reaction toward a distinct set of effectors. IL-4 promotes IgE production by B cells and may contribute to granuloma formation in response to parasitic antigens. IL-5 recruits and activates eosinophils. Remember that eosinophil link because it's central to the ocular parasitic disease we'll cover shortly. So where is Th2-mediated DH important? In parasitic infections, in late-phase allergic reactions, in asthma, and in atopic dermatitis and other atopic diseases. Notice the through line, IgE and eosinophils, the hallmarks of allergy and antiparasitic immunity. This is the same Th1 versus Th2 seesaw we introduced early in the lecture, now playing out at the level of tissue inflammation. Th1 giving macrophage-rich granulomas, Th2 giving eosinophil-rich, IgE-associated reactions. When we reach ocular toxocariasis, this Th2 pattern will be exactly what drives the eosinophilic ocular granuloma.
There's a third pattern of delayed hypersensitivity worth isolating, the granulomatous form. When intracellular organisms or extracellular parasites persist and can't be cleared, the immune system may wall them off, producing granulomatous DH. Its typical features are induration, granuloma formation, and multinucleated giant cells, the same architecture we saw building in the tuberculosis example. But here's an important clinical caution. Granulomatous inflammation isn't always a clean antigen-specific T-cell response. It can also result from immune complex deposition, from heavy metals, from foreign body reactions, and even from innate immune activation without any clearly defined antigen-specific T-cell response at all. That's a humbling point, and the slide makes it explicit. In many autoimmune diseases, the exact contribution of specific T-cell pathways remains incompletely defined. So when you see a granuloma histologically, resist the urge to assume a single mechanism. Keep that nuance in mind as we now move from mechanism to disease, examining a specific, well-characterized example of Th1-driven ocular autoimmunity, sympathetic ophthalmia.
Sympathetic ophthalmia is one of the most instructive diseases in all of ocular immunology, so let's give it proper attention. Clinically, it's a bilateral granulomatous panuveitis that can follow penetrating ocular trauma to one eye, with the fellow, sympathizing eye, becoming inflamed. What makes it so valuable pedagogically is that it's a rare example in which an autoimmune disease can be linked to a specific initiating event, the injury. Several mechanisms are proposed for how trauma breaks tolerance. First, exposure of previously sequestered uveal autoantigens, self-proteins the immune system normally never sees. Second, primary immune sensitization at conjunctival or extraocular sites, where antigens are presented in a less protected environment. And third, disruption of the normally suppressive immune environment of the retina, RPE, in uvea, the eye's immune privilege being breached. Together, these changes permit an intraocular immune response to overcome the local regulatory mechanisms that usually keep the eye quiet. Next, we'll look at the specific effector arm that does the damage.
So, which effector mechanism actually drives the inflammation in sympathetic ophthalmia? The response is thought to be dominated by Th1-mediated delayed hypersensitivity directed against uveal or retinal antigens, exactly the Th1-Dh pattern we built up 2 slides ago, now aimed at the patient's own eye. The evidence fits that picture on several fronts. CD4-positive T lymphocytes predominate early in the disease course, consistent with a helper T-driven Class II restricted process. Activated macrophages are prominent within the granulomas, the expected fingerprint of interferon gamma-driven macrophage activation. And Th1 cytokines have actually been identified in the vitreous of affected patients, direct molecular confirmation. This is a satisfying convergence of clinical picture, histology, and cytokine data, all pointing to one mechanism. And it's not an isolated case. Similar Th1-mediated mechanisms are implicated in several other forms of ocular inflammation, which is why understanding this prototype pays dividends across uveitis. Now, for contrast, let's swing to the opposite pole, a Th2-driven ocular disease, ocular toxocariasis.
For a clean contrast with the Th1 story of sympathetic ophthalmia, consider ocular toxocariasis, a Th2-driven disease. Toxocara canis is a nematode acquired by ingesting viable eggs, which then mature into larvae in the intestine. The initial immune response begins in the gut, and it mounts a strong Th2 response, precisely the polarization we'd expect against a parasite. The primary effectors are exactly the Th2 signature we discussed, IgM, IgG, and IgE antibodies, together with delayed hypersensitivity T lymphocytes. Now the ocular part. A small number of larvae may evade this immunity and spread hematogenously to the choroid or retina, eventually invading the retina or vitreous. Once there, local Th2 cytokines recruit eosinophils and macrophages, producing the characteristic eosinophilic ocular granuloma, the lesion that defines the disease. Notice how every element traces back to the Th2 pathway: IgE, eosinophils, parasite. Hold that in mind because the next slide dissects the antibody-dependent mechanism by which those eosinophils actually injure the larva and the surrounding tissue. Let's zoom in on precisely how tissue injury happens in ocular toxocariasis, because it's a beautiful example of antibody and cells cooperating. Anti-larval B lymphocytes may infiltrate the eye and produce immunoglobulins locally, particularly IgE, the Th2 signature antibody. That IgE, or IgG, then binds parasite antigens displayed on the larval surface, effectively tagging the larva. Now the effector suck. Eosinophils attach to those bound antibodies through their Fc receptors, the Fc region serving as the docking handle, exactly as our structure-function discussion predicted. Once anchored, the eosinophils release cytotoxic granules containing antiparasitic cationic proteins right next to the larva. The result is targeted damage to the parasite, but also collateral inflammation of ocular tissue. Mechanistically, this resembles antibody-dependent cellular cytotoxicity, or ADCC, a combined mechanism we'll formally define later. The elegant point is the division of labor. The antibody provides specificity, the eosinophil provides the weaponry. It's a preview of the combined effector responses that close out this lecture.
We now turn to the second major type of lymphocyte-mediated response, the cytotoxic T lymphocyte, or CTL. These are antigen-specific CD8-positive T cells, specialized for killing, and their principal targets are virus-infected cells and tumor cells, cells whose interiors have gone wrong. They can also contribute to graft rejection and to some autoimmune diseases. Recall our HLA discussion. CTLs read HLA class 1, so their preferred targets are cells displaying intracellular proteins, including viral antigens, through class 1 molecules. That's the surveillance of the cell interior concept made lethal. Two important requirements. Full CTL differentiation often requires help, signals from CD4-positive helper T cells, and local helper T cells and co-stimulatory molecules may also be needed for maximal target cell killing. So even these direct assassins depend on the helper network we've emphasized throughout. On the next slide, we'll get into the actual weapons, the two distinct molecular mechanisms by which a CTL destroys its target.
How does a cytotoxic T lymphocyte actually kill? Through two principal mechanisms, and the slide gives them memorable names. The first is direct lysis. Think of it as assassination. The CTL releases perforin, a molecule that inserts into the target cell membrane and forms pores. The resulting loss of membrane integrity causes osmotic cell lysis, and the target essentially bursts. The second mechanism is apoptosis. Think of it as inducing the target to commit suicide. The CTL can trigger the target's own death program in two ways, through Fas ligand, FAL, or CD95L, binding to Fas, CD95, on the target cell, or through cytokines such as TNF. Rather than blowing the cell apart, this quietly instructs it to die in an orderly fashion. And here's a clinically elegant feature. CTLs generally produce limited lymphocytic infiltration and can kill their targets with relatively little surrounding inflammation. That precision, killing cleanly without a big inflammatory mess, distinguishes CTL-mediated killing from the macrophage-rich chaos of delayed hypersensitivity.
This figure illustrates the two CTL killing pathways side by side, and comparing them visually cements the distinction. On one side, the perforin route. The CTL docks onto its target, releases perforin, and you see pores punched into the membrane, water rushing in, and the cell lysing, the assassination pathway. On the other side, the apoptosis route. Fas ligand on the CTL, engaging Fas on the target, or TNF delivering the signal, and the target quietly dismantling itself through programmed cell death, the induced suicide pathway. Notice what both share: exquisite antigen specificity via HLA class I recognition and minimal bystander inflammation. That's the visual takeaway: targeted, contained lethality. Keep this image in mind as we transition to the next effector, because natural killer cells use very similar cytotoxic weaponry, perforin and apoptosis induction, but arrive at their targets by a completely different logic, one that doesn't require antigen specificity at all. That contrast between the CTL and the NK cell is where we're headed next.
Natural killer cells are a fascinating counterpoint to the cytotoxic T lymphocyte. NK cells are non-T, non-B lymphocytes that kill the same kinds of targets, virus-infected cells and tumor cells, using cytotoxic mechanisms similar to CTLs, the perforin and apoptosis machinery we just saw. But there's a crucial difference. They lack antigen-specific receptors, and unlike CTLs, they don't require prior priming or activation. They're ready to kill from the outset, hence natural. So how do they choose targets without a specific receptor? Through a balance between activating and inhibitory receptors. Activating signals promote killing of cells that lack normal HLA class I expression, or that display abnormal, inappropriate surface molecules. Meanwhile, inhibitory receptors recognize the expected host cell ligands, normal HLA class I, and prevent the destruction of healthy tissue. This is the elegant missing self concept. NK cells kill cells that have lost their normal self markers, which is exactly the trick many viruses and tumors use to hide from CTLs. NK cells close that loophole.
We now reach the third and final category from our three-part scheme, the combined mechanisms, where antibody and cells cooperate. The prototype is antibody-dependent cellular cytotoxicity, or ADCC, which we previewed in ocular toxocariasis. Here's the logic. Antibodies bind antigens on a target cell, but in this case they may not directly cause complement-mediated lysis. Binding alone doesn't finish the job. Instead, effector leukocytes recognize the exposed antibody Fc region through their Fc receptors, docking onto the antibody-coded target. That interaction then activates the effector cell's cytotoxic machinery: degranulation, cytokine release, and target cell killing. Here's the conceptual heart of ADCC, and it's worth stating precisely. The effector cell itself is non-specific. It has no idea what it's attacking, while the antigen specificity is provided entirely by the bound antibody. So it's a partnership. The antibody aims, the cell fires. That division of labor between a specific targeting antibody and a non-specific killer cell is the defining feature of every combined mechanism we'll discuss.
Let's fill in which cells perform ADCC and where it matters clinically. A range of effector leukocytes can mediate it: NK cells, macrophages, neutrophils, certain T lymphocytes, and, importantly for the eye, eosinophils through an IgE-dependent mechanism. That last one should ring a bell. It's precisely the eosinophil and IgE partnership we described attacking Toxocara larvae in the eye. Where does ADCC contribute overall? To tumor immune surveillance, to antimicrobial defense, to graft rejection, and to some autoimmune diseases, a broad and important portfolio. But the slide offers an honest uveitis-specific caveat worth remembering. ADCC is probably not a major mechanism in uveitis itself, though it may contribute to antiparasitic ocular inflammation. So calibrate its importance appropriately for your practice, central to antiparasitic responses like toxocariasis, but a minor player in most uveitic disease. With ADCC covered, we move to the last combined mechanism, and arguably the most dramatic, acute IgE-mediated mast cell degranulation.
Our final effector mechanism is the fastest of them all, acute IgE-mediated mast cell degranulation, better known as immediate hypersensitivity. Here's how it's set up. Mast cells bind IgE through high-affinity Fc receptors, and critically, they bind it by the Fc region, leaving the antigen-binding Fab arms pointing outward, exposed and ready. The mast cell is now armed, waiting. When a specific allergen arrives and cross-links two adjacent IgE molecules on the surface, that bridging event triggers rapid mast cell degranulation. And rapid is the operative word. This reaction occurs within minutes, which is why it goes by the names immediate hypersensitivity, type 1 hypersensitivity, and, in its systemic form, anaphylaxis. When the mast cell fires, it releases two categories of substances: preformed mediators stored ready in its granules, and newly synthesized inflammatory mediators generated on demand. We'll catalog both on the next slide, because knowing which mediators are involved explains both the clinical signs and the drugs we use to treat them.
Let's catalog the mast cell arsenal, because these molecules explain both the symptoms and the therapeutics of allergic disease. The preformed mediators, stored in granules and released instantly, include histamine, the star of the show, serotonin, heparin-containing proteoglycans, the proteases tryptase and chymase, which incidentally serve as useful markers of mast cell activation, and chemotactic factors that summon eosinophils, neutrophils, and monocytes to the scene. Then, generated fresh on demand, come the newly synthesized mediators, prostaglandin D2, leukotrienes, and thromboxane, all arachidonic acid derivatives, along with a suite of cytokines, IL-4, IL-5, and IL-13, plus TNF-alpha, IL-1, and the chemokine CCL2. Notice something important in that cytokine list: IL-4 and IL-5 again, the Th2 cytokines. The mast cell doesn't just cause an acute burst, it actively reinforces the Th2, allergic, eosinophilic program we've traced throughout. That's the molecular bridge to the sustained, late-phase reaction. On the next slide, we'll translate this mediator list into the clinical effects you actually see.
Let's translate all those mediators into what you actually observe clinically, and then close the loop on treatment. The mast cell mediators produce vasodilation and increased vascular permeability, contraction of bronchial and gastrointestinal smooth muscle, and increased mucus secretion, the physiological core of an allergic reaction. Beyond that immediate burst, the mast cell's cytokines recruit eosinophils and other inflammatory cells, sustaining the late-phase allergic response, so there's an early wave and a delayed, smoldering second wave. Clinically, this manifests across a spectrum: erythema, urticaria, and angioedema on the skin, airway obstruction in the lungs, and, at its most severe, plasma leakage and hypotensive shock, anaphylaxis, that can be life-threatening. And here's the therapeutic payoff that ties it together. The benefit of antihistamines in acute allergic reactions highlights the central role of histamine among all these mediators. Block that one preformed molecule, and you blunt much of the reaction, a direct line from the mechanism we've built to the drug in your clinic.
And that brings us to the end of Chapter 2. Let's step back and see the shape of what we've built. We began with the defining features of adaptive immunity: antigen specificity and memory, and the vocabulary of antigens and epitopes. We then walked the immune response arc, phase by phase. the afferent capture and HLA-restricted presentation of antigen, the processing phase, where helper T cells differentiate into Th1, Th2, Th17, and regulatory subsets that dictate everything downstream, and the effector phase, where antibodies and cytotoxic cells eliminate the threat. We saw memory turn a sluggish primary response into a swift secondary one, and we grounded every mechanism in real ocular disease: sympathetic ophthalmia as Th1 autoimmunity, toxocariosis as Th2 parasitic granuloma, Fuchs' uveitis revealed by local antibody production. Carry forward this central idea: the helper T cell subset that dominates determines the disease you see, and increasingly, the therapy you choose. Thank you for your attention.